ladies and gentlemen, we are pleased to have with us Dr. Henning Hammajakob. Dr. Henning joins us from EMBL, European Bioinformatics Institute, Cambridge. Dr. Henning, after having completed his MSc in Bioinformatics, joined the European Bioinformatics Institute in 1997, where he leads the Molecular Systems Cluster. He had joint appointment as Director of Bioinformatics at the National Center for Protein Sciences, Beijing, from 2015 to 2019. Dr. Henning's team provides a broad portfolio of resources for systems biology, ranging from molecular interactions, INACT, and curated pathways, Reactome, to systems biology models, biomodels, at the highest level of abstraction. Supporting the domain-specific resources, his team develops broad-scope data infrastructure resources, identifiers.org, and the Omics Discovery Index. As founding member and SC member of the Proteomics Standards Initiative and the SAP member in Systems and Translational Molecular Biology projects, Dr. Henning contributes to the international standardization of data representation in molecular biology and the implementation of community standards in high availability, stable public resources. With these words, we welcome Dr. Henning. Thank you.
All right, let's welcome Dr. Henning for his session. He is already there with us, and I'm sure he does not need much introduction. All of you have been hearing about him. So over to you, Henning. Please, you may share your screen and take over the session now.
Okay, first of all, thanks for the nice introduction. And it seems I am not allowed to share my screen, although I thought I had set it up.
We can see your PPT. We can see your PowerPoint.
You can see it. Okay, that's great.
Just make it full screen, please. That's it.
I have no idea why I get the error message, but anyway, so you see now the presentation mode?
It is nice, yeah.
Okay, great. So thanks a lot for the very nice introduction. And by the way, I am not necessarily the one who created Reactome. I'm one of 4 Reactome co-PIs. I'll come back to that in the final slide. But I have been with Reactome for a rather long time, since about 2007. So at least I have contributed a lot to it, perhaps. Okay, so I'm honored to be able to present here what we are offering in Reactome, and I'll try to focus on the analysis capabilities, and how to then get access to the results, but I'll also briefly give a bit of background. So Reactome's aim is to provide what we call computationally accessible pathway reviews. So basically to turn text like the one on the left here, simple journal statement on what's going on in one pathway, and take not only one of these, but many, many of these, and turn them into a computational representation in Reactome from a very high level, visually attractive overview, all the way to detailed molecular maps where you see every individual reaction and reactant in a pathway. The standard building block of Reactome is a reaction. And for us, reactions are both classic biochemical reactions with inputs, outputs, and catalysts, but also other types of biochemical events like binding, dissociation, degradation, phosphorylation, dephosphorylation, transport, etc. All of these are reactions which connect by their shared inputs and outputs into pathways. But what is important to keep in mind is that pathways are a human construct. Biochemical reactions are real, but pathways are just linear slices through very complex biochemical networks, which make it easier for us to understand what's going on. But it's important that they are only one view into a complex network of events which all happen at the same time or to different time schedules. And we try to make that accessible in Reactome by linking from one reaction to other related ones between different pathways, etc. Reactome for human is now the largest pathway resource, at least in the public domain, and annotates more than 11,000 protein coding genes. If we take about 20K human protein coding genes, we cover 11K. We extend the scope of Reactome by high confidence interactors from the INTECT database of molecular interactions. And then there's quite a large space of proteins where we will probably never be able to put them into Reactome, simply because there's nothing known and progress in assigning new functions is quite slow. So there is quite a large gap where it simply makes no sense to put things into a biochemical pathway database. So the space of remaining curation, at least based on the protein level, is actually not that big anymore. But of course, there's often many events a protein is involved in. There's much more than just proteins. There's RNAs, secondary metabolites, and so on. So we are still far from complete coverage, but we have a very good coverage, at least of the human proteome. Reactome curation is entirely manual. For every pathway we curate, we recruit a domain expert. The expert then works with our curator. And when both are happy with the representation of a pathway in the database, then we recruit another domain expert as an external reviewer. So really the process is very similar to what's happening in writing a journal review, which of course results in a fairly slow, but we believe also very high quality process. Now a bit more on what you actually see when you use Reactome. This is the core view of Reactome, the pathway browser, which on the left-hand side shows the hierarchical view of the interconnected pathways in Reactome. The hierarchy goes from very high-level pathways like cell-cell communication via multiple levels of subpathways to eventually the individual reactions which form part of this pathway. Then in the main viewport, you see the detailed molecular map, which is fully zoomable. When you zoom out, like here, you see highlighted subpathways through these colored boxes. When you zoom in, you see more molecular detail. In the bottom, you have various tabs which provide more information on whatever you have selected in the main window. For example, here, the manual textual description of the pathway, which is curated by the domain expert. This is not text mined from somewhere, but explicitly curated. And as I said, you can zoom in and get increasing levels of detail. Like here, you see a part of a molecular map where functionally relevant phosphorylations are annotated, where the detailed reactants and reactions are shown, and you can zoom in even further. I'll show that in a couple of later slides. Now, this course is focused on proteomics, and so a key functionality of Reactome is to provide pathway analysis, where you can use your experimental results from high throughput experiments, whether it's proteomics or other approaches. In the simplest case, you can simply use a list of genes which are interesting to you by whatever definition. For example, those which are enriched by at least twofold, and you can paste these into the Reactome analysis window, or you can provide more complex data. Like here, we are showing multiple columns of quantitative data. In this case, this is data from the HPA, Matthias Uhlén's protein atlas, and it shows on a per tissue level, expression levels for the respective protein as non, medium, or high. And these are then analyzed. We always perform, at least in the main Reactome web application, a standard gene enrichment analysis to show which pathways are enriched in the data set you have provided. And then you get an overview like this one, where on a genome-wide view of Reactome, we show what's happening in your data set with respect to the Reactome annotated pathway space. And you can see here the enriched pathways based on a color map. And you see each of the top level bursts in this view, which we call the fireworks, is a top level pathway, and then it goes down into the branches of the pathway, which are the sub pathways. And you can see this can be multiple levels, down to about 7 or so levels. And while this view provides a quick high-level overview, sometimes you have to quite squint to see what's actually going on. And so we have more recently added an additional view. If you click on this spider web-like symbol in the fireworks overview, you get to what we call Reactome, which is a visualization of the same data based on Voronoi maps. So each top-level pathway here is represented as one top-level bubble. And this representation is space filling, so we have many more pixels per pathway to color. Therefore, you get probably much clearer representation of the data. The cell size represents the pathway size. So you see the largest pathways in Reactome are metabolism and immune system. And the color represents, again, the overrepresentation as mentioned before. But we can also, if you provide, like I showed in this example, if you provide quantitative values, we determine not only the overrepresentation, but we also then calculate the average value per pathway, per stage, so per column. And we map this to a color map, so you can also see this. I'll show that a bit later again. You can zoom in by double-clicking on each of these cells, or by doing a long press. You can then zoom into the standard Reactome pathway browser. And if you did this on a top-level pathway, then you see our textbook-style high-level pathway diagrams. We have now complete coverage of these for all the top-level pathways, except a few which are missing for the disease area. And these are custom-made representations drawn by a biomedical illustrator. And they are SVGs, they are not PNGs, so they are arbitrarily zoomable without getting pixelated. And they are completely free for you to download. They are editable in tools like Inkscape, and you're completely free to use them in your own illustrations. They also serve not only to give a quick overview of what's going on in this top-level pathway, but also they serve as a table of contents. And so if you click on any of the labels, then you get to the next lower level, or in the pathway hierarchy. So you can zoom in gradually from here on the left, cytokines in the immune system, to different types of signaling processes, of signaling molecule classes. And you can zoom in further and see more detail. And because these are SVGs, we can also modify them computationally. And we are overlaying information on these. For example, here, the results of the pathway analysis I showed earlier. So here you can see that in the subpathway signaling by interleukins, 135 genes or proteins were in your data set out of the 1,225 which are annotated in this pathway. Then you can zoom in further and you get to the detailed molecular maps, which I've already briefly introduced, where you see the individual reactions, the reactants, and small chemical entities. And you can see again the data which you provided mapped onto this molecular map. The colors here then represent the quantitative values you provided. As I mentioned, we show then on the detailed view, we show for each protein an expression value on this map based on what you have provided for this protein or gene. For this tissue, at the bottom, there's a bar which allows you to tap through the different tissues and then observe how expression changes in the different tissues. We are overlaying quite a lot of external data on Reactome. You can, for example, for any pathway which is selected in the main window, you can click on the expression tab and you see for all the genes which are attached to whatever you've selected in the main window, you see the expression values from a number of reference gene expression experiments from the expression atlas, so that you can directly compare your data and reference data from the expression atlas. I've already mentioned we are using in high and medium confidence interactors from the intact interaction database to extend the scope, the coverage of Reactome. And whenever we have an interaction which has one protein annotated in Reactome, then we show the interaction anchored on this annotated protein Reactome. And for each protein in Reactome, you can see these little red baubles in the upper right corner, which gives the number of interactors associated with this protein. And if you click on that bauble, then this is expanded and you get the view of the interactors in a kind of halo around the protein. If you zoom in enough, then both on annotated proteins and on interactors, you see little PDB structural pictograms for each of these proteins so that you can get a quick view of what the structure is like. And if you click on the interactors, you get a link to UniProt to get more information. If you click on the edge, which connects the two, you get information, you get linked to intact, and you get more details on how this interaction has been determined. Another way of using this feature is to overlay other types of information. And this is the only part which I'm mentioning in the next 3 slides, which is still experimental. So we are using DisGenet as an example of overlaying other binary information. DisGenet provides gene disease associations, and we are pre-processing these relationships from DisGenet into a simple table, which we can then use almost in the same way like protein interactions. One side is a protein, the other is the disease name. And then we can use this as an overlay And we can show for every protein annotated in Reactome a set of diseases associated with this protein or with mutations in this protein. And this works also for multiple proteins at the same time. So you can get views like this, where you can see that some disease associations are shared by multiple proteins in one pathway, while others are unique to that specific protein. And in the same way, you could overlay, let's say, biochemical reagents as pathway modulators or drugs. So it's a very flexible system to provide additional information anchored to Reactome. So this was the view where we show many diseases associated with one gene. For DisGenet, we also show, and this is already in production mode, the other way around, where we show for one disease all the genes associated with it. And the URL is given here. It's also a part of the Reactome menu tree now. So we have, for each of the diseases in DisGenet, we have generated the list of all the diseases associated with it, of all the genes associated with it. And then you can choose one of interest, like let's say here, breast carcinoma. And then we run the standard around clicking the button analysis. We run the standard gene set enrichment analysis, and you can see in all the Reactome views I've presented before, which are the pathway areas which are strongly related with this particular disease phenotype. In some cases, it doesn't make much sense to do an enrichment analysis if you have only a handful of genes associated with a disease. In this case, we can change from showing the p-value to showing only coverage. So here you see for a disease which has much less associations, just those pathways where at least one gene is associated with the disease, and then the color map still shows what is the coverage of the genes in this pathway based on the disease associations. And this can be simply switched in the menu at the bottom of this view. Throughout the talk, if you access the talk from which I've uploaded, there's many more slides which provide more detail, which I've cut out here for time reasons, but also I've put in a couple of exercises. If you want to do these exercises and you get stuck, feel free to send an email to Reactome help, and we will then help you and guide you further along the path in these exercises. But I don't show them here for time reasons. Okay.
Would you like participant to do something, Professor Henning?
Sorry?
Would you like participants to do some analysis on? We have given them some data actually today. It was actually, interestingly, some COVID data which we did on mass spec. And one of our team members showed them some network analysis. Would you like them to try out on Reactome and see what they get?
Yes, of course. So they are very welcome to try out Reactome with their data sets or follow the things which I've put in here. And if there are questions, just send me an email. I've put in my email at the beginning of the presentation, or send an email to Reactome help, and we are happy to answer questions if there are any problems. So yes, please.
That's right. So in fact, what I would suggest to participant, if you have any specific question for the presentation so far, you can ask to Professor Henning, or if you want to try out the data which we have provided you, just try to go through that same data and analyze in Reactome. And then if you have any specific query, then we can also do that. It will just hardly take you 2 minutes time to do the analysis in Reactome. So whichever way you want. Any questions in the meantime from participants? Is everybody able to follow all the steps? Looks very clear.
Hello, sir.
Yes. Professor Kapil?
I think many of us would like to do the analysis of the provided data using Reactome. So if just Professor Henning could guide us right now, then it would be very helpful.
Right.
I have no idea what exactly the data is which has been provided. So I think it might be better if you do that and then ask concrete questions.
Right.
Okay, so that would be better.
Arup, can you please chip in here and just show the data? And maybe you can just walk through with the Reactome to the participant to show that data.
Yeah, I can do that.
Then they can start analyzing, and then their queries can be taken by Professor Henning.
Sure, that would be great.
Participants, please be ready to share your screen to show the output, and any specific queries or more detailed understanding of that can be taken with Professor Henning.
Right. So
I guess I have to unshare then, right? Oh no, it seems to work already. Okay.
right. So participants, this part is clear, right? These were this is the raw data that you have. This is the fold change value. This is the t-test. So any questions up till here? After that, you have to open reactome.org. So actually, so this will be the first page that you will be landing. This is the landing page. Then click on Analyze Data. Then it's pretty simple. You go ahead, paste. So you paste the entire and click Continue. Then you have to, since it is a human, you can just say Project to Human and click on Analyze. So thereafter, I don't know, you guys can follow? Then you have to go to Download. Then you can click on the Pathway Analysis Report. Then you will get a CSV file as a result output. And if you want a really detailed report of how which were the pathways were enriched, you can just click on the PDF file. So a PDF will open.
Can you please go back to the downloads? I mean, after downloads, this is not coming. After analysis.
So this, you are on this page, right?
Yes. So this part has come, but what happens?
Right. This is the results file. So basically,
Yes.
these are the pathways that got enriched. Okay. This shows the not found. These are the, as I told, like sometimes there are few IDs that cannot be mapped. So you have 27 IDs that could not be mapped. And then if you click on downloads.
Okay, on the left-hand side. Okay, great.
One is the CSV file. Like even in Metascape, you had seen that you need to know, right, which were the proteins which are mapping onto which pathways. So that CSV file is basically Excel. It will open with Excel. Then you will see these are the pathway IDs. So metabolism of RNA, and you will get all the details. What were our submitted? Which were the proteins that we submitted that came up here, right? And then you will find a PDF file, right? You have to click on PDF, and depending on your browser integration, either it will come up as a separate download or it will open in a new window as well. So once you open the PDF file, you will get a detailed report that Reactome has just generated. So that will have an introduction, and then you would find genome-wide overview. Okay, so some error here. So basically you get then these are the most significant pathways. And once you click on the pathways, it will take you to a very wonderful representation of how these pathway works and which are the, I think Professor Henning can take it from here.
Well, I guess that's already the main. So you basically, it shows the top 25 most overrepresented pathways. And depending on whether these are top level pathways like we had just before, then you get the detail, let's say, textbook style illustration. And if you select a pathway which is further down in the list, you get the detailed molecular map. But a key point in these PDFs is that for both representations, the resolution is set to very high, and that means that you can basically keep this pathway analysis and still use it for your publication a couple of months or even years later. This responds to a request we had where people said, I have done my pathway analysis 1 year ago, and now I want to write my publication, but everything has changed, or things have changed, which I was interested in, which is normal because Reactome changes, gets updated. But once you have downloaded this pathway analysis, then the visualizations are in high enough resolution that you can actually continue or that you can use them in your own publications, and therefore you have a kind of archive of your analysis results of that version of Reactome against which you did the analysis. And then as you see here, you get the entities which are found in this pathway, you get the editing history, and so we try to provide all the information which might be relevant for this particular pathway analysis even a while later, so that you're independent of versioning. Of course, if you are happy, if you use your results right away, then you don't really need the PDF, because we also have downloads of all the data for the analysis as you have run it right now. This is really more if you want to archive your results. So what I've shown so far is all, in terms of analysis, is all based on simple gene set enrichment analysis. So it's basically qualitative analysis. We are, if you provide quantitative values, we are visualizing them on the various Reactome visualizations, but the analysis itself is essentially qualitative. It only takes into account the genes, not really their quantitative expression values. The advantage is this is very easy to do. It's fast. You have seen you can really do the analysis live in a couple of seconds interactively, and you just need a list of identifiers. So it's very straightforward to get a quick first overview of what's going on in your data set. But of course, we are completely ignoring, except for coloring, we completely ignore quantitative information, which means there's reduced statistical power. You have to do the thresholding outside in your own calculation rather than within Reactome. You have to do a separate analysis for up and down regulated genes and proteins. So for a real in-depth analysis, you really want to go deeper, and you want to really do a quantitative analysis. And that's what we are providing with Reactome GSA, which has been recently published in MCP. And Reactome GSA does the analysis on the pathway level, which means that we have a fairly straightforward way to do comparative analysis between multiple omics types. In one simple analysis run, one single analysis run, you can compare proteomics, RNA-seq, microarray data. You can even do an analysis of single cell data. And for all of this, we have not redeveloped new algorithms. We are just using the power of R. We are using well-established R-based algorithms and use R for the calculations in the background, of course, with the Reactome data. And then you can choose if you want to do subsequent visualizations like the ones I've shown here at the bottom in R, so stay in the R world, or if you want to use Reactome as a kind of high-end visualization engine and see the results of your analysis in all the representations with the data overlaid like I've shown earlier on. In any case, we are, thanks to a couple of let's say convenience functions which we are providing. This is work all which has been done by Johannes Griss from the University of Vienna. You can really, with a few lines of R code, you can do a fairly complex Reactome analysis, assuming you already have your data in R, and can get then quite detailed results out. This is all available as a bioconductor package, but it's also accessible directly from the Reactome web interface, which then hands you off to R, and you get the results back in the web interface, so you can access the functionality from both worlds, from the Reactome web interface and from R. In Reactome, it then looks something like this, where you can see, again, on the detailed molecular maps, the up or down regulation for the data you have provided. And as I mentioned, you can combine multiple omics types. And you can see, for example, like I hope you can see here the cursor in the bottom right corner, that in the data sets you've used, for some pathways, the regulation, the upregulation or downregulation goes in different directions. Like for the third pathway here, mitochondrial protein import, based on the proteomics data, there's a strong upregulation. Based on RNA-seq data for the same sample, there's a downregulation. So you can really see how trends are different in different omics data sets you have provided. And again, all of this is available for download to make accessible and to reuse in various forms. And you can cycle through the different results, like here, this is proteomics, this is the RNA-seq data, and you can change the visualization, going through the multiple columns of data you've provided. Of course, in this case, the analysis itself takes a bit longer and can take between a minute and 20 minutes, because the underlying quantitative analysis is much more expensive computationally. All the data which we are providing, all the visualizations, we make as easily accessible as possible for further use. You can download the visualizations as SVGs or PNGs, as shown here, including the overlay of your data. You can choose the resolution and the format. For the detailed molecular maps, we even provide a PowerPoint download. And this is not a GIF or PNG, so a pixel image embedded in PowerPoint, but these are PowerPoint objects which are connected. So you can download this representation and then, for example, quickly move objects out of the way to make space to highlight your results and what you have is relevant to you within the existing Reactome visualization. We've already just seen the PDF report, so I don't need to go into more detail there. One point which is relevant beyond pure pathway analysis is what we call the Reactome icon library, where based on the SVGs, the textbook images I've shown earlier, we are collecting the repeated elements from these textbook elements, diagrams like cell elements, cell types, receptors, and so on. And all of these we collect as individual icons, which again you can download in a variety of formats and freely use in your own visualizations, grant proposals, PhD thesis, presentations, whatever. And they are completely free. The only thing we request is that you cite us. So this is the standard CC BY license. And again, these are vector images, so they are easily scalable and modifiable to adjust to your own purposes. So I've mentioned CC BY. Reactome is entirely open source, open data, open graphics. You can reuse everything. Just please cite us. Some content is even available under CC0 for Wikidata, but even there, we appreciate if you cite us. And the software components have been written from the start so that they are integratable into local pipelines. And we are documenting how to use these different software components. And we are not only saying you can, but we are also providing a list of external partners which reuse these software components. So they are actually being used in third-party sites. For example, in the Encyclopedia of Proteome Dynamics, where they use the Reactome widget in their application to show what their proteomes look like on Reactome space. For Reactome content, we are always interested in collaborations to do targeted curation for specific topics. We have done these recently on interleukins, on autophagy. We are working on pathogens with Cabana, a large network in Latin America, with UCLA on cardiovascular drug action, and with OICR on cancer. I'll skip the cite us because I've mentioned that already, but on every page in Reactome, you get a little cite us button, which gives the right reference to cite. And they may, of course, be different depending on the context. One thing I would like to briefly mention is disease annotation. Normally, Reactome annotates the normal reaction in a generalized generic cell, but we do quite a lot of disease-specific annotation, which is an active research topic on how to represent this well. But I just briefly mention here the three main types, how we do this. One is loss of function. This is typically metabolic disease, where one protein might be broken, one enzyme, and then the pre-products accumulate, or the products are missing, and this is what is causing the phenotype. Here you see a nice example from cystic fibrosis, where we show how CFTR mutants cause part of a reaction to be broken, and then how the drug Ifcaftor can be used to stabilize a broken complex and re-establish this reaction. It doesn't always work this nice, but this is a particularly nice example. Gain of function is typically in cancer, where for example, some receptor is constitutively active and then causes a lot of pathway activity which shouldn't be there. We overlay these additional reaction chains then on the original diagram, which sometimes makes for a difficult to read, very complex diagram. And that's why I said we are still working on trying to work out the best way how to represent such complex maps, where already the base map is complex, and then you have a complex disease overlay. And then eventually we annotate host pathogen interactions, which are more like normal diagrams, with the only difference that the pathogen proteins are highlighted in red, like other disease-involved molecular entities in our maps. And of course, then also for disease, we do high-level diagrams, like here we've done in a kind of disease curation marathon for SARS-CoV-1 and SARS-CoV-2 over the course of the last year, basically. And with that, I'd like to finish, react and do the acknowledgements. As I mentioned earlier, Reactome is a multi-site collaboration. The PIs are Lincoln Stein at OICR Toronto as the lead PI, and co-PIs Peter D'Eustachio in New York, and Guanming Wu in Portland, Oregon, and myself at DVI. We have an outstation in Beijing, and we have quite a large team of curators, developers, and external collaborators. The quantitative analysis Reactome GSA, which I mentioned, was largely done by Johannes Gris. And all of this work would not be possible without funding, which in our case comes mainly from the US NIH, from EMBL core funding, and from some BBSRC national funding, and from the public-private open targets collaboration. And with that, questions or more questions?
I'm sure all of you who have done the analysis will really admire that how beautifully this works. In a very short time, you can get wealth of information from this reactome. Anybody wants to ask any question? All right, Dr. Piyali Mukherjee, please go ahead.
Thank you, Professor. This was really interesting to see this. I was just curious because I have in the past used ingenuity pathway analysis for these kind of interactome. How different do you think, or if you can explain how different or how precise this is the reactome analysis would be from that? Or is there a difference?
Okay. Obviously, my view is biased, but the first point is that Reactome is entirely free. So you can do anything with it. You can reuse the entire Reactome data in your applications. There's no restrictions on any further distribution. So, and obviously it doesn't cost money. That's the other point. It's completely free. And so apart from that, I believe that Reactome has a very high quality because we have this very detailed, let's say, journal-style review process with 2 independent domain experts which are contributing to the pathway content. And for human, at least, we have a very good coverage. But we focus on curation of human data. So for other species, we do orthology-based inference, and that obviously works well for other mammals. That works less well if it goes along long evolutionary distances to non-mammals. And so I can't really judge if ingenuity is better for non-mammalian species because they have simply bigger coverage. And that's basically my perspective. So I believe that for human, I would recommend to use Reactome. For others, probably you're better off using Ingenuity or other resources like KEGG. But given that I don't have Ingenuity available, I can't directly do the comparison.
Thank you, Professor Hemming. That's really helpful.
That's a very candid comment from Professor Hemming. And given that it is such open access, software available, and you can very quickly analyze your entire data set with high quality, I think that's really makes quite a big edge on the using Reactome versus anything else. One participant, Dr. Hemant Gowda, had asked, what is the specialty of Reactome? It's really nice to use software. Is there anything you want to ask in terms of the specificity? But I think that Professor Henning already had mentioned that it is quite rigorous peer review process which they go through it. They will do the manual curation data analysis which goes through peer review process. So what data pathway output you get, that is actually quite stringent in that way. So I think that probably has already been addressed. Sushmita, you want to ask some question?
Yeah. Hello, sir. So my question is, when we are giving a list of proteins as a input file in Reactome and then analyze those, want to analyze the pathway for those proteins, few of the proteins are not mapped in Reactome. But even we see that those proteins are very much significant in our data analysis in terms of the statistical tests. So how to like address those proteins, or how we get in what pathways those proteins are involved in.
So, as I mentioned before, Reactome has a good coverage, but it's not perfect. And the first thing I would recommend is that you try the tick box include interactors. So on the second page, as soon as you have submitted your data for analysis, there is one page which has basically two options. One is project to human. As I mentioned, we focus on human annotation, so we provide the capability to project from whatever, let's say you have used pig identifiers to project to human just because we have better coverage in human. That's one. But the other one, which is relevant here, is then include interactors, and that will extend the scope of Reactome by about 50%, and this will catch many more participants in the reactions and take them into account in the analysis. But of course, then also the underlying statistics change. So you also analyze against a larger background distribution, which may be good or not, but you have to keep that in mind when you interpret the results. But even then, it's perfectly possible that some proteins which are significantly regulated in your data are not annotated in Reactome. It's not complete, it's not perfect, like any biology resource. And that's basically as it is. And the best, if you know about this protein, if you really want to address it, perhaps not only one protein, but a whole area, we are very interested, as I mentioned in some of the last slides, in working together with a community to increase our curation. So please do tell us if you don't find in Reactome some proteins which you consider important. Ideally, even if these are proteins of your expertise, we would love to work with you on getting them properly annotated. But if that is not an option, well, there are always some which are not included in the analysis because they're simply not yet covered. That's inevitable almost if you have a large enough data set. On the other hand, in the pathway analysis, you would hope that your effect is touching more than one protein. So hopefully that because a pathway is relatively broad, you will still see the right pathways coming up, even if a few proteins are not covered in the analysis.
All right, I think, yeah, this is the right way of moving forward. If I think some participants are having the proteins which they are quite keen to understand and they don't see them in Reactome, they can contact Professor Henning, and I think his team might be able to curate better. All right, so well, I suggest all the participants to take your data sets and play with Reactome and try to add interactors and see are you able to get interesting biology out of your data. And in case if you have some query, please also email Professor Henning, and he will be, I'm sure, able to look at your request and probably reply back to you. With this, Professor Henning, let me thank you wholeheartedly for accepting our invitation and giving this wonderful lecture, some demonstrations. And participants are, I think, toward the end of this week, they'll be more comfortable taking their data from mass spec and microarrays and doing the pathway and network analysis. And everybody is going to use Reactome for sure. So thank you so much for the wonderful work and the presentation.